Hello, cell biologists. This is Caleb.
This is Michaela.
This is Alina.
And this is Brendan. We're the HeLa group, and we're presenting on the pulse chase assay.
The pulse chase assay is useful in cell bio for two main reasons. The first reason is it allows cell biologists to understand cellular processes as they occur over time in the cell. The second form of the pulse chase assay, which we will not be examining in depth during this presentation, but encourage you to research for yourself, is finding the half-life of molecules such as proteins or mRNA. Two notable examples of the pulse chase assays used in cell biology are George Palladi's charting of the secretory pathway and proving the existence of Okazaki fragments. In cell biology, cellular processes occur in separate places and over periods of time.
The pulse chase assay is divided into three stages, with the first part being the pulse phase. The pulse phase begins when a large dose of radioactively labeled molecule, in this case the amino acid leucine, is added to a cell's culture medium. So as you can see in this picture, we have our molecule, which is the amino acid leucine, that is radioactively labeled here, which is represented by the red triangle, and a large dose of it is being added to this cell's culture medium. And so radioactive amino acids are thereafter incorporated into the proteins that are manufactured during protein synthesis. So as you can see in the second picture here, we have the protein that's being synthesized that is incorporating those radioactively labeled amino acids into it, and that is represented again by the red triangle. The second part of this assay can be referred to as the chase phase, which begins when a large amount of a non-radioactively labeled molecule, again here we have the amino acid leucine, is added to the sample. And so as you can see, the only change from the last slide is that this amino acid leucine no longer has that red triangle that was representing the radioactive label. And so essentially we've added the same compound to the sample, but the non-radioactively labeled version of it. And now at this point, the radioactively labeled proteins are no longer being made, which you can see from this second picture that the protein being synthesized no longer has those red triangles that were representing the radioactively labeled amino acid.
In order to track the progression of radiolabeled molecules through the cell is fixed at various time points. Fixing the cells essentially freezes all molecules in place, including the radiolabeled proteins. In this slide, radioactively labeled molecules are visible as black spots after staining in microscopy. Because the samples are fixed at various time points, the progression of proteins through the cell can be determined. These images show how radioactively labeled leucine was tracked through the rough ER to the Golgi apparatus and eventually secreted from the cells and vesicles.
The pulse chase method can be better understood if compared to a popular research strategy in conservation biology, which is GPS tracking. This technology allows conservation biologists to understand animals' physiology and behavior and interactions between humans and animals. One example of GPS tracking can be seen with the recovery of the California condor, where conservationists successfully reintroduced condors back into the population using winged GPS trackers and have been monitoring their population ever since. Another example is how UC Santa Cruz has partnered with the U.S. Fish and Wildlife Service to monitor puma populations in Northern California. Using GPS collar tags, researchers can better understand their interactions with other pumas, prey, and other humans. Both examples are similar to how labeled protein can be introduced into a cell to see where that protein goes and how that protein interacts with other parts of the cell.
Now that we're familiar with pulse chase method and how it works, we're going to see it being applied to an actual experiment. This paper is from JACS, or Journal of the American Chemical Society. The article is trying out new labeling probes that have a wider application, and one way that they test the probes is through pulse chase analysis. One of the experiments they wanted to conduct is to see whether one can track the internalization of target proteins and cell surface expression with differentiation. Some older probes normally wouldn't be able to monitor such a quick process, and the new probes allow for both real-time tracking and the ability to track different things in the same experiment. To start the experiment, epidermal growth factor receptor proteins found on cell membranes were labeled red with a probe called RA. The cells were allowed to culture, and then another label, FCA-PO2, this time green, was added to the cells. Two important things to note is that RA is membrane impermeable, and FCA-PO2 turns green shortly after interacting with BL-tagged proteins. This process was visualized with the confocal fluorescence microscope, which allows for 3D imaging of a process with pretty high resolution. So what was observed? At first, they only saw the red fluorescence, and with time that started to decrease, and as the red fluorescence decreased, green fluorescence started increasing. Let's put those results into context. The red fluorescence present at the beginning of the experiment diminished with time. So if the red probe is membrane impermeable, that means that endocytosis of the red electron growth factor receptor proteins was responsible for the decreasing intensity of the red fluorescence. After the red started diminishing, the green fluorescence started appearing. This indicates that the epidermal growth factor receptor proteins that were engulfed by the cell have started expressing identical proteins labeled green, and those proteins are being sent to the cell membrane surface. This is a visualization of the engulfed proteins expressing identical proteins that are being sent to the cell membrane surface. Looking down at E, we see that at 0 minutes, there's barely any green present. They image the cells every 2 minutes, and we see the green fluorescence increase with time. This further confirms that the previously red proteins that were engulfed by the cell started expressing new proteins tagged green that were being sent out to the cell surface. This shows us one of the ways that we can use pulse chase analysis to keep track of protein movement, expression, and half-life.